Okay, this is continuation of spinocerebellar tract, and for this pathway you don't really have to draw the pathway with the scenario, but this is just to help you to appreciate the wirings of the pathways. So cerebellar pathways had that three things in common: the input, goes first to the cerebellar cortex, and the next is deep cerebellar nuclei, and that's the output of the cerebellum. Well, the only exception to that circuit is the vestibulocerebellar circuit, which doesn't go to the deep cerebellar nuclei, but goes straight to the vestibular nucleus. So we can apply this to spinocerebellar pathway. So input, we had two inputs, but we're just gonna use the Mozzie fiber for this illustration. So Mozzie fiber is the input, and goes to the cerebellar cortex. And for the spinocerebellar pathway, the cortex that it goes to is either vermis or paravermis. And the deep nuclei, cerebellar nuclei, is vestigial nucleus or interposed nucleus. And the output is to the upper motor neurons. This example is showing the relationship between this tract and the lateral upper motor neurons, and you can see the person is trying to move the arm, which is the distal muscle of the arm, and because it's distal, the pathway we're going to use will involve paravermis and interposed nucleus. Sorry for the typo here, interposed. Ter is missing here. Interposed nucleus. So let's look at the input first. So these are the cerebellum in the template, left and right, and we can see that there's outside, that's the cortex, cerebellar cortex, and inside there's some deep nuclei. So it's labeled D, I, and F. So the most lateral will be dentate, and most medial will be vestigial, and in between will be interposed nucleus. So this I is what we're going to use because we're moving the distal muscle. So this is interposed nucleus. And we can see the input and output of the cerebellum, and that finishes at the start of the upper motor neurons to move the limbs accordingly. So let's have a look at the inputs first. So the inputs we're going to look at is the Mozzie fiber. So Mozzie fiber for the spinocerebellum tract has two different types. So these two are the Mozzie fibers. Two types, one is called high fidelity fiber and the other one is called internal feedback fiber. High fidelity fiber delivers the current sensory state of the limb. So we can imagine this is which goes to the dorsal root and shares the dorsal root, and must be the high fidelity fibers. So we can label that on the right as our high fidelity tract. That forms one of the Mozzie fibers, and the other one is relaying the motor signal, which is situated near the ventral horn. So ventral horn is where the upper motor neuron is synapsing with the lower motor neuron, and because of the location of this neuron, the cell soma, it's literally eavesdropping the communication between the upper motor neuron and the lower motor neuron. So this is related to a motor signal, current motor signal, and this tract is internal feedback tract. So we can say this contains motor signal, current motor signal, and this contains the current sensory signal. So those two informations are fed into the cortex, and the cortex, it will stop, or synapses with, will be our paravermis. So this portion of the cerebellar cortex is our paravermis. And if we go into the details, the muscle fiber will synapse first in the granule cell layer, and the granule cells will send this axon, which is called parallel fibers, to the Purkinje cell, and Purkinje cell is the output of the cerebellar cortex. So in other words, this red neuron that sends the output to the deep nuclei is our Purkinje cell, or Purkinje fiber. So that's the output of the cerebral cortex. It goes to the deep nuclei, which was our interposed nucleus. And from interposed nucleus, it's the output of the cerebellum. So the word outputs came twice when I described this. Outputs of the cerebral cortex is the Purkinje fiber. Outputs of the cerebellum itself is our deep nucleus. So this interposed nucleus will send the outputs to the upper motor neurons. So the purple fiber stops at this point. This is our red nucleus in the midbrain. And that's our upper motor neuron, lateral upper motor neuron, start of the cerebrospinal tract. Cerebrospinal, which is our lateral upper motor neuron. The other fiber, if we want to activate the lateral corticospinal tract, it has to go to the cortex, and it doesn't have the direct connection. It has to go via thalamus. So this is the thalamus, and there's different parts of the thalamus that were used in the ascending tracts. It's called ventrolateral nucleus. Of the thalamus, or in short, VL thalamus. So this thalamus relays the signal from the cerebellum to the cerebrum. So it goes to the corresponding hemisphere, which controls the forearm muscle, and from that it sends the lateral corticospinal tract to contract the muscle. One thing you'll realize is that both of the fibers that goes to the red nucleus and also to the lateral corticospinal tract crosses over to the other side. And in order to see why, it becomes obvious because both of the tracts are contralateral. In other words, if you want to activate the sorry, the side is wrong. This is right side and this is left side. If you want to contract your right arm, which was this forearm, right arm, what you have to activate in the hemisphere is the opposite hemisphere, which is the left hemisphere. So this left hemisphere, neurons of the left hemisphere has to be activated in order to control the right arm. And because the cerebellum is ipsilateral, the fibers, output fibers, has to be crossed over to the other side to contract the other, activate the other part of the hemisphere. So because of this wiring, let's say you have damage to the paravermis, so you some of the neurons died. And what will happen as a result is all these signals cannot be processed and the output cannot be calculated accordingly. So you will have trouble controlling the right arm. So this is the right side of the cerebellar damage causing the right side of the dysfunction of the right arm. And the cerebellar lesions is called ipsilateral lesion. So damage to the cerebellum results in the signs and symptoms of the same limb, same side of the limb. And the reason why it's ipsilateral is because of the inputs are not crossing over to the other side and fed directly onto the same side of the cerebellum. Okay, so this is the spinal cerebellum, which is responsible for coordinating the ongoing movement. So this is ongoing movement, controlling the ongoing movement. So getting the timing right for contracting the antagonist and agonist muscle, for example, to reach the target. Our last pathway is called cerebrocerebellar pathway, and it also follows the same sequence. There's an input, it goes to the cerebellar cortex first, it goes to the deep nuclei, and goes to the output. And input is from cerebrum, as the name implies, the cerebro. So it starts from the big brain, goes to the cerebellum, and goes back to the big brain. So it forms like a loop. And because it has to go through the big brain to the small brain, it has to go through the bridge, which is known as pons in Latin. So this cerebrocerebellar pathway has to go via pons. And the function is thought to be related to motor planning. So it's different from controlling the ongoing movement. It's planning ahead of, let's say, skilled movement or well-learned movement. So when you're about to perform those movements, this pathway is activated and getting ready for the upper motor neuron to dispatch the planned commands of the movement, for the movement. So you can imagine this pathway will be light up before the movement, while the spinocerebellar pathway, it will be light up during the movement. So the input, as I said before, it's from the cerebrum, and specifically it's the cortex that mainly motor or sensory cortex. And then input goes via pons. And we can say these inputs also Mozzie fibers. So these Mozzie fibers will go to the cerebral cortex, and this green area, color-coded green area, is our lateral hemisphere. Of the cerebellum. The cerebellar nuclei is the one that is laterally located, which is the teeth-like structure dentate nucleus. And from dentate nucleus, the output will go straight into the cortex cerebral cortex. Specifically, it controls the fine motor control or well-learned skill area. So lateral upper motor neuron, and for this case, for our case, it'll be motor cortex, primary motor cortex. And this diagrammatic representation of this pathway is shown here. So the starting point is our cortex. So this green neuron, the cortical neuron, will go down to pons, and you can see this neuron synapses with the neuron in the pons, and this particular neurons in pons are called pontine nuclei, a group of cell bodies in pons, and this pontine nuclei will be the input to the cerebellum. So this neuron input will feed into the cortex first, which is our lateral hemisphere, and from lateral hemisphere the Mozzie fiber will synapse with the granule cells. Granule cells will synapse with the Purkinje cell, Purkinje fiber. So this is Purkinje fiber, which is the output of the cerebellum to the dentate nucleus. So this is our Purkinje fiber. And this nuclei is our deep cerebellar nuclei, which is our dentate nucleus. And the output will go to the cortex back, cerebral cortex back. So from the deep nuclei, the output has to go again via VL thalamus. So this is our VL thalamus, similar to the spinocerebellar pathway. It goes through the VL nuclei, ventrolateral nuclei of the thalamus. And goes to the cortex. And from the cortex, the blue line indicates the lateral corticospinal tract. So from the cortex, this information will be linked to this neuron, and this neuron will dispatch the information, cross over to the other side, and synapse with the lower motor neuron. So this is our lateral upper motor neuron, which is our lateral corticospinal tract, and this is our lower motor neuron, let's say for the arm. So we'll go through the ventral root and synapse with the muscle that controls the arm movement. So again, you can see this is ipsilateral wiring between the cerebellum and the muscle that needs the control from the cerebellum. It's the same side. So this is left side, and that's also left side. No, sorry, this is right side. Getting the sides wrong now. Right side and right side. So again, if you damage, for example, lateral hemisphere, then all the planned commands cannot go through to facilitate the lateral upper motor neurons, which will result in trouble controlling the same side of the arm eventually. So this damage will affect the right side arm muscle, and again this is indicating the ipsilateral signs and symptoms of the damage. Not only the motor function, but it's also known recently that cerebellum has contribution to perception or cognitive functions. So for example, if the subject was asked to move their fingers and try to grade the roughness of the surface, they also the researchers also reported the activation of the cerebrocerebellar pathways related to the fingertip area of the homunculus of both cerebellar and cerebral areas, as well as the deep cerebellar nuclei. So what happens if you damage this area? So you will have trouble planning the well-learned movement. And for example, if you have lateral hemisphere lesion, this will result in decomposition of movement. Or also known as asynergia. So the movement becomes a bit jerky and decomposed, and one example will be the symptoms called dysdiadochokinesia. So I'll just spell it out, it's quite long symptom. Dysdiadochokinesia. So this is one of the example of decomposition of movement, and what it shows is when you ask the subject or patients to pronate and supinate the arm, so the patients have to supinate and pronate the arm at the hand as fast as they can, so you just back and forth, flip and flip back. They have hard time doing this movement fast. And because this planning is disrupted, the brain is having hard time when we're to attune to the timing of the antagonist and agonist muscle contraction to flip the hand in fast motion. So this is one example, dysdiadochokinesia, supination, fast supination and pronation of the arm is difficult for them. Okay, lastly, we're going to cover about the cerebellar learning. And this motor learning that occurs in the cerebellum can be summarized as trial and error type learning. Trial and error type. So if one damages the cerebellum, the person will have hard time learning from the error, which is very crucial in motor learning in general. And the role of detecting the error is coming from the climbing fiber. So the climbing fibers, we haven't really talked much about climbing fibers for our pathways, but it has important contribution to give the cerebellum the information about how the error is made and how to correct it. The climbing fibers, we know that it's originating from the inferior olivary nucleus. Inferior olivary nucleus in the open medulla. So it's type olivary. Nucleus, and conveys the error signal from somatosensory, visual or cerebral cortex. And the function of this climbing fiber is the LTD. LTD is called long-term depression. Of certain synapse that causes the error. So the role of the climbing fibers is it detects the error and it goes synapse with the Purkinje cell, but the synapse is a sort of inhibitory that inhibits the Purkinje cell, which causes the error causing movement. So this depression of the synapse specifically called parallel Purkinje cell synapse. So it inhibits this synapse or depresses the synapse. Can last for a minute, 2 hours. And while it's suppressed, the other Purkinje cell outputs might be tried to find the optimal movement to do the task. So let's see how, in detail, how this is suppressed and how it can be causing the trial and error type of movement learning. So this diagram is showing how the priming fibers and mosaic fibers and related circuits are related in long-term depression. One thing to note is that we have these two different types of microcomplex shown in this diagram. And microcomplex is a group of neurons that is related, and it's independent to other microcomplexes. So microcomplex comprises of mosaic fibers and parallel fibers, and it's Purkinje fibers and climbing fibers. So in this diagram you see two microcomplexes. One, let's see, I'll just name MC1. You see MC1 here with this is mosaic fiber, and let's say this parallel fiber is part of the MC1, and this Purkinje fiber is part of the MC1, and this climbing fiber is part of the MC1. So those are the smallest unit that behaves independently. You can see other microcomplex. So let's label this microcomplex 2. So that's microcomplex 2, also including the deep nuclei as well, the neurons in the deep nuclei. So this is the neurons in the deep nuclei. So that's also MC1, and that's the nucleus for MC2. That's Purkinje fiber for MC2, and that's the parallel fiber for MC2, and that's the climbing fiber for MC2. So let's say at the moment MC1 is activated. So MC2 is silent, but MC1 is activated, and it will produce the output from the deep nuclei and activate the upper motor neuron, contract the muscle as a result. But this is creating the error. Let's say this has created the error. What happens next is the microcomplex 1 climbing fiber will be activated, detecting that this movement has created the error. So this is MC1 climbing fiber will be activated, delivering the error signal. And once the action potential travels up, what it'll do is it will depress or inhibit the synapse between the parallel fibers of the MC1 and the Purkinje cell of the MC1, microcomplex 1. And as a result, the microcomplex 1 for deep nuclei will be suppressed. So it will no longer be active, which means the output to the upper motor neuron will be suppressed. And meanwhile, other microcomplex will be activated to try out different types of movement. So now what happens is this microcomplex 2 will send the output to the upper motor neuron, and this MC2, the Purkinje fiber for this MC2 will be activated, so as the parallel fibers for this MC2 will be activated. So once this works, that's fine, but once it doesn't work, the same pattern will repeat it again. So once it creates the error, the climbing fiber for MC2 will be activated, suppressing this synapse for a few minutes or a few hours, while letting the other microcomplexes to be tried out. So in these mechanisms, we shows that the cerebellar is able to try different options of the movement while suppressing the error-causing types of movement. The evidence of this motor learning type, trial and error type, is directly coming from the patient's case who damaged the climbing fibers, the inferior olivary nucleus. And this study is best exemplified by the PRISM study. And PRISM study, the subjects wearing a PRISM goggles. And what these PRISM goggles do is, normally if we see so this is, let's say this eye, if we face straight forward, the gaze is actually parallel to the head. But with this prism glass, what happens is it tilts the gaze. So the prism deflects the light and the gaze becomes tilted, even our head isn't straight. So what you'll see is the scene from this side, not from the front side. If you're not really sure how this works, the best example would be this prism glass reading prism glasses. So you can see the woman is wearing these prism glasses, and the light is deflected in 90 degrees, so she will be able to read a book while resting on the bed. So this example is just tilting the light sideways, not in this this picture that I just showed you. So this study looked at the throwing little balls at the target. And for normal people, when they first wear the prism glass, so let's have a look at the graphs first. So on the left hand side, this is the normal population. On the right hand side, that's the patients with the inferior olivary nucleus damage. So if you see the left hand side, there's a time on the x-axis, so it's before wearing the prism glass, and after wearing the prism glasses. And you'll see the displacement of the ball in relation to the target, and 0 means the bullseye, straight to the target, and the deviation to the right or left. So with a few trials, well, even without wearing prism glasses, they're fairly accurate in hitting the bullseye. Once they wear the prism glass, this is the first trial that they wore the prism glasses, and it deviates from the target by -50 to the left. And the reason why this happens is, even though you're facing forward, so this is showing an example of that, the purple arrow shows the throw direction and the blue is showing the gaze direction. So before wearing the prism glasses, it's all aligned, and once you wear the prism glasses, the gaze deflects to the left, and your tendency to do compensate for that deflection is throw in the same direction as the gaze. And because you're throwing at the same gaze direction, the ball goes to the left with the same gaze deflection angle. So it deviates a lot from the target. But interestingly, within 10 to 50 trials, they adapt to it. So it's the trial and error type learning. They realize which direction would be nearer to the target, and eventually they learn how to throw at the target with the deflected gaze direction. So what happens after the learning, the throw direction becomes straight, so straight to the target, while the gaze is deflected by wearing the prism glasses. So this angle is learned. What's interesting is that if you if the subject take off their glasses, they don't normally go back straight to the normal state. What they do is they maintain this relationship, this learned relationship, which is this angle. So they will see the target straight, but they will throw the ball to the right because they learn this angle will lead the ball to the target. So ironically, the first try they throw is actually veered to the right, which is opposite the first time they're wearing the glasses. And within few trials, like 10 to 30 trials, they get back to normal, so they learn the relationship between the gaze and the throw direction. So this is also known as after effect. Where the adaptation, learned adaptation persists after the trial. But all these involves the trial and error type learning, and this is possible due to the intact learning mechanisms of the cerebellum. We can say that because if you see the patients with inferior olivary nucleus damage, so this is showing you the upper medulla, and that's the area where the patients had damage to, and that area contains the inferior olivary nucleus. And their pattern is, so this is before the prism glasses, and this is during the prism glasses, and that's after taking off their prism glasses. Most of the time they're hitting the bullseye before the prism, wearing the prism glasses. And when they wear the prism glasses, the gaze direction and throw direction is aligned, and they tend to throw to the left, and they never get to the bullseye compared to the normal population. So the adaptation doesn't occur for these patients. They just keep throwing at the towards the left of the target. When they take off the prism glasses, the after effects is not really exaggerated. They just keep hitting the bullseye right after taking the prism glasses. So you can see, compared to the normal population, there's no or lack of evidence of the learning through trial and error, and this is mainly due to the climbing fibers, the role of the climbing fibers, which is to deliver the error signal to the cerebellum and try out the different microcomplexes. And this is impaired for these patients, and not really seeing that trial and error type learning here. Okay, so in the next video, I'm going to just talk a little bit about the cartoon that I made.